much for the opportunity to show our data here. My name is David Brenner. I'm from Ulm University, from the Neurology department, and I would like to talk about NEC1. First want to start with overview over the genetics of ALS in Germany. And you see from this graph, so it's on the one side is displayed genetics in familial ALS, and on the other genetics in sporadic ALS. And you can appreciate that genetics play a role in both ALS forms. So 8% in the familial cases, and in the sporadic patients in Germany, it's around 16%. And among the most frequent affected genes by variants of unclear significance is NEC1. And that's why we asked, so how could we find a solution to be able to classify these variants into benign or pathogenics? And this is closely linked to the knowledge of the mechanism. So at the moment, we only know that truncating variants are associated with ALS. No missense variants have been found to cause ALS in a monogenic or Mendelian way. So there's no pedigrees with co-segregation. We know that there's a polymorphism that is associated with ALS, but it increases the risk. It's not a cause of ALS per se. And it's also not yet known how this polymorphism is linked to ALS exactly, in which direction also. And but ultimately, a Mendelian missense variant could be the solution to find the pathomechanism that underlies NEC1 ALS, if one would compare it with a truncating mutation. Well, however, such a missense variant was not available up to now. We were very lucky to find such a variant in routine testing of a patient who turned out to be familial, who had several affected siblings from ALS or suspected ALS, and they all were positive for this variant. That is the PN598S variant in NEC1. Parents were not affected, but so we know from NEC1, from truncating variants, that it's rather the rule than the exception that there's incomplete penetrance, like actually for most haploinsufficiency genes, even for SOD1 and C9, you know. I think the more we test, the more we see that the most of the ALS genes are incompletely penetrant, and it depends also on the genetic background of the families. But so we had a look at the conservation of this variant, and this is only a small glimpse of species where it actually is conserved. I could make the list much longer, and you see it's conserved and also the surrounding. It actually is located in the coiled domain of the NEC1 protein. And we next ask, is there maybe even an enrichment in ALS of this specific missense variant? Because so far, a missense variant as a group have not been found enriched in ALS, but that's the problem, that you cannot throw all missense variant in one pot, because this is just not adequate. You can do it with the truncating variants because they're all the same effect, but you cannot do it with the missense variants where you have some a proportion that are pathogenic and a proportion that are not. Yeah. So and actually you dilute the probability to find actually pathogenic variants if you put them in the same bowl. So we just checked for this variant and fused two large cohorts, the German MND-Net and the Project Mine, and analyzed the burden of this variant in both cohorts combined, and we see that there's a significant enrichment if you can compare it to the European part of the GNOME AD data set. So another hint that this variant is very interesting to look at. The most important thing was actually that we had access to a autopsy. So we were lucky, and that such a such rare variant autopsy is available, but it was. And we checked protein expression in ZNS tissue here, and found that this variant has a normal expression. So and this is in compared to a truncating one. Yeah? Otherwise, it would have been boring. Yeah, and the variant. So we also looked at TDP43 pathology and P62 pathology, and you see, can appreciate that it leads to TDP43 pathology. This was already known from Jenna Gregory's lab, that NEC1 ALS is associated with TDP43 pathology. But what was not known, that it is also going along with P62 pathology, and actually more prominently in our missense variant than in the truncating variant. But so we will follow up this further in neuropathological analysis. That's just a small excerpt. But we were not, I think we were not satisfied, and went into IPC-derived motor neurons to further explore the consequences of this missense variant and to compare it with a truncating variant. And we generated isogenic motor neurons to be really sure that what we see comes from the genotype. And you can see here again that the protein expression is lost in a truncating variant, but not in our missense variant. And then we looked for axon pathology by staining for neurofilament in this chamber setup, in this fluided chamber setup, where you have the axon and the neurons separated, and that's why you can really nicely quantify the axons. And you see here that it's the wild type, and these are the two NEC1 genotypes. So there's a axon pathology. And then we looked for an apoptosis array, and you see there's a clear separation between the wild type and the two NEC mutants. And interestingly, it's more pro-apoptotic in the missense variant, what might hint to a dominant negative effect, but that's just speculation. And then we looked at DNA damage, because NEC1 is involved in DNA damage repair. And we saw that there's also higher susceptibility in the NEC1 genotypes, and actually that is even a bit higher in the missense variant. And the last phenotype we checked was also already known from other studies, is cilia. So ciliary morphology, and it's known that truncating variants lead to shorter primary cilia, and this was also the case for the missense variant. I don't show a quantification here. So in summary, our missense variant could elicit similar phenotypes in IPC-derived motor neurons that had previously already been shown for truncating variants. And the next was that we followed up the neuropathology results. We looked for P62 accumulation, and this was also the case in the IPC-derived motor neurons, also in the NEC1 mutant, in the missense mutant ones. And we looked for TDP43 mislocalization, and we could also discover TDP43 mislocalization in both NEC1 mutants, both in the truncating and in the missense variant. And most interestingly, we could rescue this mislocalization by overexpressing NEC1. What I think clearly shows that there's a link between localization of TDP43 and NEC1, what we have to explore in further studies. So I think the evident thing was to look for kinase activity next in this missense variant, because this has been hypothesized actually to be the disease-defining function of NEC1, but it had not been shown in vivo. It just was an in vitro derived hypothesis, and And that's why we looked for a known direct. So for a known direct target, phosphor target of NEC1, and this was phosphoYAP1. That's actually a very interesting protein because it has been implicated also with regulation of TTP43 localization in the recent past, and we see that phosphorylation goes down in both NEC1 mutants, and we then also isolated looked for isolated kinase activity of our missense mutant in the panel B and C, and also saw that it goes down, and we could also mimic the phenotypes we saw, the P62 accumulation, the DNA damage, and the cilia phenotype by inhibiting NEC1 with inhibitor, and so that's actually showed that what we found is due to reduced kinase activity. We also had a more thorough view at this by doing omics, so phosphoproteomics and RNA sequencing, and I just showed actually I think the summary of this results. You see that there is no simple overlap between NEC1 truncating variant and the missense variant. There is an overlap, but it's small, and that's why we are hypothesizing that there is actually also dominant negative effect by our missense variant, and it's not just a simple loss of kinase activity, but for sure it impairs kinase activity. So, and with that, I'm already at the end of my presentation, and I think we could show that heterozygous impairment of kinase function is sufficient to cause ALS. And actually in our family with a higher penetrance that truncating variants, that pathological phenotypes in NEC1 ALS, at least in vitro, are downstream of NEC1 kinase dysfunction, and that NEC1 is linked to localization of TTP43, but we have to show actually the mechanisms behind that. And we have some evidence that our missense variant might be even more penetrant and maybe not be dominant negative. But I think the most important thing is that we now have a first, we have indication or is evidence that if you have a loss of protein expression or you have a dysfunction of kinase activity, that this can trigger ALS, and that would be a way actually to classify missense variants of uncertain significance by functional testing of kinase activity. And so that's the next step, that we want to set up testing of kinase activity in fibroblasts or other cells. We have to see which cell type is the best one to do that, because that's a huge burden of genetic testing when you find this variants of uncertain significance and have to communicate these results to the families or to the patients, and they don't know if they get the disease or not, or relatives don't know if they get the disease or not. And I think this would be important to be able to make a conclusive statement here. So with that, thank you very much for your attention, and I'm happy to take questions.
Thank you for your talk. I'm here. When we look at the population database like NOMAD, the loss of functions, there is a certain loss of function variants that are they are high frequency, ALS frequency, and it's not compatible with the dominant inheritance. So I wanted to know what do you think about this loss of function variants, and if you have an explanation on why there is such a high frequency.
There's such a high frequency because it's not always penetrance. So as I already said in the beginning, a non-penetrance is rather the rule. Genes than for other genes, but also for other ALS genes. And I think it's between a Mendelian gene and a very high risk factor, and it depends on the genetic surroundings, on the genetic background of the patients, if it becomes penetrant or not, and also on the lifestyle. For example, the family with this missense variant, they are all heavy smokers, so every day one complete package. And I think this, for a gene that regulates DNA damage, that must be, I think, must matter. And I think that might be the reason, why you have non-penetrance. And it's the clear rule of all the truncating variants that there's incomplete penetrance that skips several generations sometimes.
Yeah, but in genetic diagnostics, we have the responsibility for pre-symptomatic testing also, and so we differentiate risk factors from really Mendelian mutation, and that's why in France, the 3 laboratories, we were really careful, and we discussed about that, and because of the frequency, we actually consider even loss of function variant as a risk factor.
Yeah, I think there's a good reason to do like that. But on the other hand, it's how you see it. I think it's more than a risk factor, especially combined with lifestyle. And I think you could at least recommend relatives who are carriers of this variant that they modify their lifestyle, do not smoke, or be as healthy as possible. I think there's also a positive thing about knowing that. And also the other thing is, it's such a big advantage to know the genotypes of these patients now. Now we see how incompletely penetrant most of the ALS genes are. That's important knowledge, and it's good to have these genotypes to make cohorts, to make natural history studies of these patients. And that's why I'm still happy that we keep on doing or keeping this gene in our panels in Germany. But I absolutely agree with you. You can also see it from the other side, that it's a burden for the patients to tell them, yeah.
Yes, thank you for your presentation. Based on your results, did you look at coming back to the single nucleotide variation of this gene, which was reported in ALS?
You mean
but missense mutation only?
Yeah, you mean the
to see whether it's close
R261H.
to the mutation that you identify, whether the minor allele frequency is very low, and whether software to predict pathogenicity is, bring new variant, missense variant, as responsible for ALS.
So you're referring to the R261H variant that increases the risk of ALS?
No.
Are you referring to
the
to prediction, to in silico prediction?
With prediction with respect to the position of your variant.
Yeah.
With respect to the minor allele frequency and prediction of pathogenicity based on Fred score and all these things.
So this variant, actually, you find it 30 times in GNOME-D. Some people would say, oh, it's too high actually to be pathogenic, but I think this is not an indicator. Could also be a founder effect there, and maybe you also have in a GNOME-D some people who will develop ALS. So I think it's hard to say it from this. And the other thing is, so we had also in silico prediction, there are some algorithms that classify it as likely pathogenic. And if you do an analysis with alpha missense, it will tell you it's probably benign. So actually, I don't trust so much.
You mentioned, is it this variant or other variant which has been reported?
We have only tested this variant here, so we have.
Yes, I understand that.
Yeah, I don't get the question then.
The first slide in which, variant of unknown significance.
Yeah.
My question is whether among them, there are variants which are close to the one that
yes,
you
now I get
reported.
it. No.
Whether
Yeah.
the minor allele frequency of this variant are very low, and whether software predicting pathogenicity will bring other variant, missense variant, which is in fact are not VUS but pathogenic.
There are other variants localized in the surroundings there, but we haven't checked for that.
Right.
No, and actually this is variant also is not in the kinase domain, so it probably doesn't. It's maybe owes to the quaternary structure that it affects the kinase functions. Okay, we do not know, but I think we have the chance now with measuring the kinase activity to check other missense variants what they do. No?
Hi.
Yeah.
Thank you. A really nice talk. I just have a question. It's actually related to the previous one. You think you can try to look for the variants and split your gene across domain to see if you have increased aggregation in variants in kinase domain versus cold-coding domain versus other domain that would maybe help you to actually prioritize the one that might be doing something?
Yeah, absolutely. Great questions. I also had this idea already 1 year ago, and I also approached Alfredo Jaco Ancelli because they had done it for the SOD1 gene or for the neurofilament gene. And actually, I would love to do exactly the same. I think that would be very interesting. Yes. Yeah.